thank you everyone for being here and remotely. So I'd like to just mention about this special moment that we are having here. We are doing this in honor of the Rare Disease Day, which is celebrated tomorrow, February 29th, in more than 100 countries. There are over 6,000 rare diseases affecting at least 300 million people, or 6% of the world population. About 70% of them are genetic diseases, mostly affecting children. Yet, hundreds of maybe thousands of rare diseases remain without a known molecular cause. Lack of such basic information is one of the major obstacles hindering progress in rare disease research. Investigating this question to advance genomic medicine is at the forefront of national and international research funding agencies. Then, to talk about some of these efforts and the translational impact to affected families, we are pleased to welcome Jill Hawkins. Jill is the founder and president of the FAM177A1 Research Fund. She will provide her personal family's journey from undiagnosed children to a new foundation with robust scientific network. Her goal is to unite this community and support the development of future treatments. Jill is in person, then Jen Posey is going to talk to us remotely. And she is, Dr. Jen Posey is assistant professor and physician scientist in the Department of Molecular and Human Genetics at Baylor College of Medicine in Texas. Jen will highlight the recent discoveries of the genomics research to elucidate the genetics of rare, or GREGoR for short. GREGoR is a national research program funded by the NIDRI with the goal of linking diseases to their genetic causes, which our lab is also partner. So thank you, Jill and Jen.
Thank you, Claudia, and thank you, PNRI, for having me and for celebrating Rare Disease Day. Science does matter. It matters in ways that significantly impacted our family, and I'm honored to be able to share our journey with you today. Disclosure, I'm not a scientist or a doctor. I'm a mom set on an unexpected course that's inspired me to learn and to advocate in ways that I never imagined. So let's start at the beginning. My husband, Doug, and I had our first child, Nash, 20 years ago. He was healthy and hit all his developmental milestones as expected. A year later, we welcomed our daughter, Charlotte. She was healthy at birth. She was floppy, but she fell behind developmentally. We consulted experts and started early intervention. As Charlotte continued to fall further and further behind, she had extensive medical workup. Brain scans showed delayed myelination, but were otherwise unremarkable. We suspected seizure activity, but it wasn't captured on EEG. All her labs were normal. She had extensive genetic testing for the time. This was about 2008, before the days of exome and genome sequencing. So her chromosome testing was normal, and we ruled out known disorders like Angelman's, Prader-Willi, Fragile X. So no one had an explanation why our gorgeous little girl was struggling so much. We were alone. We longed for a diagnosis and explanation that might lead to specific treatments or a community that would understand the terrible burdens of this disease, like the disrupted sleep, the challenging behaviors, the relentless need for care, and the debilitating stress that it puts on families like ours. We were told, she's a well put together little girl. Yeah, it's probably genetic, not likely to happen again. So, given what we understood about it and our desire to experience typical development as it's supposed to happen, and in part to give our son Nash a partner in caring for his sister later in life, we decided to round out our family, and we welcomed our son Cooper in 2010. Cooper too was born healthy and just as cute as his older brother and sister, but he too fell behind developmentally. And my husband and I had a dreadful sense of deja vu. And we knew pretty early on that Cooper must have whatever it is Charlotte had. Although there was no test to confirm it, our medical team agreed with us. So we were distraught. But through the grief and an intense amount of love, we charged on, more therapies, doctor's appointments, and testing. Cooper checked all the same neurodevelopmental boxes as Charlotte, and a few bonus evil ones like congenital cataracts and early seizure onset. So what was probably genetic was now definitely genetic. And this lit a fire in me. Genetic testing had evolved in the meantime, and we were fortunate to have access to top-notch care. We were followed by Doctors Dobbins and Mirza at Seattle Children's Hospital, who enrolled us in one of their research studies. Our family had exome sequencing, but this was inconclusive. They said at the time that there was this thing called whole genome sequencing, but it was cost prohibitive, new, hard to interpret, and that they needed 5 more years in order to sequence our kids. And I said, well, I don't want to wait 5 years. I heard about the Undiagnosed Diseases Network, new program through the National Institute of Health. I applied, and we were one of the first families accepted into the Stanford location. So the kids were poked and prodded and evaluated. And 3 agonizing years later, I got a call that they had found likely pathogenic variants on a gene called FAM177A1. Back up 1 slide here. Charlotte and Cooper had compound heterozygous mutations on FAM177A1, knocking out the function of the gene. And although strikingly understudied, as it was described to me, with only 5 publications ever even referencing the gene, there was one paper out of Saudi Arabia that described a family with 4 siblings with overlapping symptoms to Charlotte and Cooper and variants on this gene. So we had found the causal gene. And a little fun fact about that paper out of Saudi Arabia, it was authored by Dr. Fauzan El-Correa, who just happens to be presenting on this seminar, Science Matters in June. I don't know if he's going to be talking about FAM177A1, but I hope he'll mention it at least. So for the non-scientists in the room, it turns out that my husband Doug and I each have a complete working copy of FAM177A1. And we have 1 copy with a variant. In our case, we each have a different deletion. We had a 50-50 chance of giving the working copy or the non-working copy to our children. And it turns out we gave both non-working copies to Charlotte Cooper. And now we know you only need 1 working copy for this gene to do its important work. So we had a 1 in 4 chance with each pregnancy to have an infected child. So after a 14-year diagnostic odyssey, we set sail from Undiagnosed Island. Mind you, we were in a ragtag ship with a really lean crew, but we were on our way. I also created a virtual lighthouse so that anyone interested in FAM177A1 could find us, and it's working. Dr. Baruch Uger, a researcher at Yale in Dr. DiCamilli's lab, was working on lipid transport. She contacted me when she discovered that FAM177A1 interacts with her protein of interest. She's now a vital part of our research network and a trusted advisor. Her paper describing the work on FAM and CPF13B is approved and awaiting publication. In addition, families from all over the world have found us, and now they know they're not alone. We are a small but growing supportive community. Earlier, I mentioned that Doug and I simply wanted a diagnosis. We'd assumed that treatments were out of reach. But due to the astounding pace of scientific discovery, treatments and cures for similar monogenetic diseases are being developed. And so I got to work. I studied and contacted the authors of every paper ever published about FAM. I cajoled my way into scientific meetings, learning, and sharing the lived experience of this disease so that their research would align with patients' needs. I talked to other rare disease warrior parents further ahead of in this journey than me and consulted with thought leaders in the rare disease world. I attended the Ultragenics Rare Disease Entrepreneurial Boot Camp to learn how to create a roadmap to a cure. I assembled a team of world-class experts to advise us as we strive to accelerate the science and move as quickly as possible from bench to bedside. And in 2001, I founded the FAM 177AM Research Fund with the mission of improving the lives of everyone impacted by this disease. And now I'm going to dig into the science because, well, it matters. And because I think it's really cool. So what do we know about FAM 177A1, related neurodevelopmental disorder? And this is a working name because this is still considered a novel disease because we don't have enough published on it. So I'm going to go off script for a second and say one of the hard truths that I've learned in this journey is the agonizingly slow pace of academic publishing. As well-intended as researchers are, it's painfully slow and frankly harmful to patients that are suffering. So we are still considered a variant of uncertain significance on genetic panels. So we're missing patients. A little bit more about that later. So we know of about 20 patients worldwide. This is undercounted for reasons I just mentioned. All patients have biallelic predicted loss of function variants on FAM 177A1. These variants include homozygous frameshift, nonsense, and exon level deletions, and shared phenotype macrocephaly, which is large head size, global developmental delay, intellectual disability, seizures, behavior abnormality, tone issues, and gait disturbance. The mechanism of this disease is unknown. And I have a couple of videos, and these are shared with permission from the families. So you can see the unusual gait that all our fam friends have. Oopsie, what happened? All right.
Yay!
Yay.
As we've connected with other families, we share images of our children's shoes who all have a similar wear pattern, that they just have this toe drag due to ataxia, and we burn through shoes at a rapid pace. And then the spinal pictures of our son's Cooper in the hospital last year. We had to make 9 trips to the hospital due to uncontrolled seizure. So the science of FAM, I'm going to call it FAM because FAM 177A1, it rolls off my tongue, but it's quite the alphabet soup. It's a highly conserved, ubiquitously expressed gene that localizes to the Golgi complex. It's found in almost every cell of the body. It's a critical immune-associated gene. It makes a protein that we now know is vital for health. It's a very small protein. Its function's undefined. And because it's small, it is amenable to an AAV therapy. RNA sequencing data and metabolomic data on patient cells and in animal models and cellular models suggest dysregulation of pathways associated with apoptosis, inflammation, and cell proliferation. So there's a lot of work to be done, but we're moving fast. This is a solvable problem, and we have a plan. Here you can see some of our research priorities. I suspect there are some brilliant scientists watching with expertise in these areas. I urge you to make a fantastic choice and study FAM 177A1. Here are some of the projects that we're already working on that demonstrate that we're a highly motivated, research-ready patient group. We have patient cells and plasma banked and ready to ship to researchers. We have zebrafish at Washington University, flies at the University of Utah, and mice at Jackson Labs. We're collecting patient data, and we are awaiting the results of a proteomic study to identify disease-specific biomarkers. The average drug takes 10 to 15 years and millions, if not billions of dollars to develop. These are resources that rare disease groups just don't have. That's why rare disease groups like ours are looking for new uses for existing drugs and doing this with high throughput drug screens. I want to tell you about our drug screen project with Dr. Clement Chow at the University of Utah. Dr. Chow has created FAM deficient flies to model the disease. The FAM deficient flies are very unhealthy, and 100% of the male flies die in the pupae before they can reach adulthood. This is called a phenotype. Then they take those flies and feed them 1,500 FDA-approved drugs, and they see which ones improve the health of the flies. You can see from this graph that most of the drugs had no effect, but many did. And we had 33 drugs that rescued the male flies more than 80%. And our top hit rescued 96% of the male flies. So these are safe, available drugs that might be effective on FAM disorder. So Dr. Chow and his team are validating those hits on some other phenotypes, including seizures. And then together with our medical team, we'll determine which drugs that we might move into FAM patients. Okay, pop quiz for all the smarty pants out there. One interesting finding from our study is that more than 60% of our top hits had a documented acetylcholine relationship, but we don't know why. One research said the biology is telling us something, but what? If you have ideas, reach out. So, in summary, after a 14-year diagnostic odyssey, we're moving fast and in a short amount of time. We're connecting and supporting families. We're building a collaborative, patient-focused research network. We hosted the inaugural FAM 177A1 Family and Research Roundtable in June. Families shared stories, and 10 lecturers gave really interesting talks from genetics to gene therapy, soup to nuts. We covered it all. You can watch that on our website, curefam.org. The bad news is, as I mentioned, we are still of us, so we're missing patients. Hopefully, with some more published literature, we'll move to the likely pathogenic category soon. There's an adage, too rare to care. So like most rare diseases, we have a solvable problem, and it's just a lack of resources. And then the ugly. This is a devastating disease. And despite being warriors and really, really cute kids, our kids are suffering. They require intensive, nonstop care, and our families are to the brink. Our daughter, Charlotte, is the oldest living patient that we know of with FAM disorder. So we don't know the prognosis of this disease. She's on a course to lose her ability to walk. Our son Cooper has regressed because of his seizures. But what if we can discover disease-modifying treatments in a time frame to keep Charlotte walking and to free Cooper of the assault on his brain? What if we can develop a gene therapy and get it into future patients in a time frame that prevents this disease altogether? What if? In conclusion, on Rare Disease Day, I want you to know that science matters. It transforms lives. And by working on rare disease, you'll impact families like ours. And you just might unlock some of the mysteries of the larger medical juggernauts, like Alzheimer's and ALS. And to the researchers, thank you. I encourage you to engage with patients. Spend time with a family impacted by rare disease. Our door is wide open. And because rare diseases are not so rare, odds are there's another rare family in your neighborhood. Take a selfie with a rare kiddo. bring it to work. And when you look up from your microscope, be reminded of why you're doing this. Science matters. The work you do matters, and you can make a difference. So on behalf of these fantastic warriors, whoop, that just disappeared. Thank you very much.
Hi, I'm Vittorio Gallo, Chief Scientific Officer at Seattle Children's. First of all, I want to thank you for this beautiful talk. It was really inspiring, and I worked at Children's National Hospital for 23 years before joining Seattle Children's last September, and the work that we do depends so much on families' undiagnosed and families' integration of the work with the researcher. So you thanked us, I need to thank you. I'm a still active researcher. My lab is downtown here in Seattle. I'm a developmental neuroscientist, and I work on neurodevelopmental disabilities and premature brain injury. So everything that you said resonated incredibly with me, and I really want to thank you. But you brought up to our attention a lot of important issues, including the issue of publishing results in a timely fashion, and which is not always under our control. Actually, most of the times it's not, because we rush into submitting articles, but then the review process. And so I think that we could work together, researchers and families, to put pressure and to bring the attention of editors and editorial boards of how important it is for families who are suffering through these issues every day to publish our work in a timely fashion. And so I think that what you said is really important, and it resonates. I have a comment on and thought on the acetylcholine question that you asked. It made me think about my early career when I was studying the effect of neurotransmitters on brain development. So there's a lot of literature on how neurotransmitters are not only involved in signaling between neural cells, but also at some specific stages of development, they can be involved in shaping neural circuits development. So I haven't caught up with the literature on acetylcholine more recently, but I think there is a possibility that at least to a certain extent, acetylcholine may have a role in shaping cell development in the brain and neural circuits development. So I think it's something to think about, and something that is an additional role for a neurotransmitter like GABA and glutamate and other neurotransmitters have in the developing brain. Something to think about.
Yeah, we will be giving it more thought, and I might connect with you to brainstorm
Absolutely. I'll
ideas.
give you my card as soon as we're done.
Great. And I love your enthusiasm and your knowledge of the field, and knowing the value that patients and families can add to research. And I love that you're here in Seattle, and I really think that this region can and should be a world leader in this area. We have the resources for
I
it.
think we're in that trajectory, and I hope that we will be #1 very soon. But I think one important point that I want to make, that you made, that even when we identify mutations in a specific gene, we are not done. I mean, it's really the beginning, because understanding, with 7,000 pediatric genetic diseases, only 5% of them have some therapeutic interventions for. But we need to understand the mechanism. So the work that you are doing, understanding the cellular mechanisms of these mutations, is really important. So thank you.
So I actually have one comment, which is that I don't think the publication timeline is usually the journals. It's actually the researchers, the clinicians who are involved, who are waiting to publish, because they want to collect more data. Sometimes it's to be sure, but a lot of times it's because they want to get into a higher profile journal. And so they're waiting until they have the mouse model or the zebrafish model or whatever, and put that together to get the paper out. But when they could have published, like, for example, I'm aware of your story. I've been following you for a number of years. You're actually mentioned in a paper we have in review right now, as an example of families who are not waiting for the publication to look for treatments. And the number of families that exist right now could be published. This could be published within 3 months. I can guarantee. One of the deputy editors for HEG Advances, published by ASHG, and I can tell you that's sort of our publication timeline. It's that people are waiting and sitting on this data. So we've actually, we created a website, MyGene2, so that families can just put information up there online and say, here's our phenotype, here's our variants. We want this to be available to everybody else so that we don't have to wait for this long, protracted publication process.
Yes.
Yeah, and I think that's something that as a field that we have to kind of recognize that we are holding this progress back because we're trying to essentially gain.
Yeah. And now that I know more about it, I understand that that is their currency and it is important. And had I known then what I know now, the importance of having a publication, I was told by GeneDx that they need at least 2 publications. They need a lot, there's a certain amount of evidence before it can move over. And they only had one, the one I referenced. And so what I would have done right away is worked with someone to capture the information that we have and somehow get it published. Call it a case study, just get something out there. But I didn't know that. So that would be something that I think that's a change that we can make, is working with these newly diagnosed families, clinician, to get something published sooner.
Yeah, the problem is that sometimes even if you publish 1 patient only or 2, it takes some time to go to databases that OMI or GARD or others. They are, so I guess we are still a little behind on that, in that sense. And that is important, right? Because of the way that the doctors oh, this disease is already recognized. Until then, they are not. So.
Right. And unless a family or a clinician might get the sequencing report back and gets the list of buses, and unless they really dig into those and say, what if it's this one, then the family goes undiagnosed. And unless they go back and revisit, they could be undiagnosed for a long time. Yeah.
I had 2. One was just a comment on this conversation with respect to kind of not only the publications, but for example, the ACMG guidelines that determine whether or not you can call something, I know you know this, but whether or not you can call something pathogenic, variant of uncertain, significant, or benign, also depend on the number of patients. So for example, our lab does functional studies, and so we have hundreds of variants that have 0 activity in our assay, but until there are X number of patients, individuals who come, present with symptoms, that's still going to be a bus. So that's just one comment. Is also just the number of patients, and kind of how ACMG needs to kind of wrestle with this stuff as well. My other question was a science question. So you have 30 compounds that look like they behave really well in flies. You also have a mouse model. Are you guys working with Jex to test these in mice?
They are willing to do that. Our mice are still being characterized. So they're viable, but they're not breeding as fast as they were hoping. They told me they're accelerating the breeding schedule, which is interesting. And they're beginning characterization. And as soon as they have a colony built out, they'll test whatever compounds we want. And we develop a gene therapy, they're going to put it in the mouse too. They've been an amazing partner for us. And our zebrafish core, they're going to test our top hits too.
Do you have any questions in the chat? If you have any questions online, maybe we can encourage people to go ahead and put those in the chat, or raise your hand if you'd like to do that.
Thank you, Jeffrey.
So first of all, thank you to Dr. Carvalho and PNRI for the opportunity to speak to this group today. It's of course being sort of rare disease, the day before the Rare Disease Day. This week, I think there are a lot of exciting activities going on at a lot of institutions, and I'm really excited to be able to be a part of the PNRI sort of celebration, if you will. And of course, I'm following a really wonderful talk. I think it's interesting to hear some of Jill's comments and to think about how excited I get at times about our research and where things are and what we've been able to accomplish. And then so helpful to really be able to put this into the context of, it's still not enough, not fast enough. There's more work to be done. And I'll echo some of those sentiments from a slightly different angle as we go forward. And I will try to keep an eye on time as well, because I think the Q&A will be the most fun part of the day. So with that, I'll be talking about our rare disease research program called GREGoR. And this is a slide I've used a lot over the years, but I think it maybe sets the tone nicely, and perhaps especially after Jill's talk, to really start with the fact that more work is needed to be done in the rare disease research world, and there are a lot of outstanding questions. One that we think about a lot is the fact that we still don't fully understand the biological functions and disease relationships of all 20,000 protein coding human genes. So these are now older data, but from the former Centers for Mendelian Genomics Consortium, which was also a rare disease research consortium, very focused on identifying novel disease genes and gene disease or gene phenotype relationships. You looked at sort of the number of novel discoveries over the eight or 8+ years of that program, you see that trajectory really kind of continued upwards with no evidence of slowing down. And for us, this was very, I guess, kind of reassuring that the impetus to continue to understand these things, that this was important, that there was more to learn, was really highlighted looking at this. But it goes a lot beyond just disease gene discovery. We need to understand the influence of different variant types, different variant locations on gene and protein function and their ultimate impact on health. And I'll speak a little bit to that, as well as the impact of combinations of pathogenic variants or damaging variants in more than one gene or locus. So the work that I'll be talking about today really comes primarily from work that we're doing here at Baylor in the GREGoR Consortium. And I think Claudia mentioned this at the very beginning, but GREGoR stands for Genomic Research to Elucidate the Genetics of Rare. We have a data coordinating center at the University of Washington, and then there are 5 research centers. The University of Washington, and I heard Jessica Chong's voice in the room, so I know she's there. I think I saw Mike Bamshad online. I'm not sure if there are others, but there are at least 2 research centers represented here today. But also Broad Children's National, which is partnered with Invitae and also now has a component at the University of California, Irvine, Stanford, and then of course the Baylor site. And before I get too far, I really wanted to acknowledge that we have an absolutely amazing team at the Baylor GREGoR site. I get to PI this program with Jim Lepsky and Richard Gibbs. I'm not sure if either of them are on today, but it's really so much more than just the 3 of us in our labs. So there are multiple other labs that are very closely involved and members of our team. Pengfei Liu and Fritz Sedlizak and their research groups. We also have a lab that's a member of our team at UT Health, kind of essentially next door, the School of Public Health at the University of Texas, that's led by Zeynep Koban-Akdemir and her research team. And then I definitely wanted to highlight Claudia. Of course, she was formerly here before she moved to the PNRI. And while I know that she collaborates with many of the GREGoR research centers, we like to try to claim her as ours. And a lot of the work that Claudia has led, I know she also collaborates with UW, and she's wonderful, and I'm sure that they would agree. She's led some really important work, and I'll touch on a small portion of that, but a lot of the work that our group has done with Claudia, or that really Claudia has done in collaboration with our team, looking at things like MECP2 complex genomic rearrangements, how that can influence phenotypic expression for those families, studying immunodeficiencies, studying some of our unsolved cases from our Turkish neurodevelopmental cohort. She's been heavily involved in some of the tool development that's happened here, looking at how to predict nonsense-mediated escape, looking at how to better identify computationally copy number variants and structural variants, how to visualize them. And then a lot of work, I will hopefully have time to touch on this, a lot of work related to genes associated with autosomal dominant Robinow syndrome, and also our understanding of the phenotypes associated with those genes. And so I just want to, it's exciting to be able to talk to this group, and thank Claudia for the opportunity, and also thank her for her tremendous collaboration.
Thank you, Jen.
Okay, so in terms of our research program, I wanted to give a little bit of an overview to start, and I'll touch upon different aspects of these, but some probably more than others. If you were to step back and ask, what are really the kind of the core aims of our research program, or how are we doing this rare disease research right now? I think that there are four main components of our program that we focused on. One is really data aggregation, and by that, I think we believe it's very, very important to get genotype and phenotype data, to get these data into a single harmonized database. I'll show you some examples later on about how important the deep phenotyping is in addition to the genomic data. And something that I kind of put here to just remind myself to mention to this group, although many of you may already know, GREGoR as a consortium is depositing our genotype and phenotype data, our genomic data, as well as metadata, family pedigree structures, a pretty elaborate data model into Anvil. And so these data are available for other scientists to apply for access and analyze. It's a very important part of the program. Another arm of our research relates to application of what I've called new technologies, which maybe not so new, some more than others. Long read sequencing technologies, RNA-seq, optical genome mapping are a few of the examples that I would say we're using a little bit more often, but certainly not limited to those. Analysis tools. I've listed a few. Dupe Finder for identifying homozygous duplications from exome data. NMD escape predictor. This is the nonsense mediated decay escape predictor that I mentioned earlier that Claudia has been very involved with. And then VisCNV is a visualization tool for copy number variants and structural variants in genome data. And members of Claudia's team have worked very closely with our team here in developing and testing that tool. And then finally, functional validation. Here, I think that comes in a few different flavors in particular. We have a branch of our team that's working on saturation genome editing and using that to then apply multiplexed assays of variant effect. So trying to understand the functional impact of some of the different variants that we're identifying. And then we also have a branch that's looking at induced neurons and what we can learn from studying particular variants, not just in any cell type, but in the cell type that's most relevant for the phenotype. We have a pretty large proportion of neurodevelopmental and neuromuscular phenotypes in our cohorts. And so induced neurons was sort of a very, very important way for us to be able to study some of the variants that we're identifying. In terms of recruitment and data generation, and I should mention that we are very much building upon the work that was started by at Baylor and Baylor Hawkins, so Jim Lepsky and Dave Valley, the Baylor Hopkins Center for Mendelian Genomics. What I'm showing you here is really what we've done that's GREGoR specific, but we have built upon the previous rare disease databases that had been developed here. So recruitment over the past almost 3 years of GREGoR, we have over 1,800 individuals, over 500 families data and reanalysis. We have sequencing data of various types on about 1,500 individuals. And we are also very interested in taking extant exome and genome data in particular, often from diagnostic labs, so patients that may have had clinical testing already, reanalyzing those data in the research setting. And so we do ingest those data from external labs, harmonize it to our data set, and analyze those as well. And so that's the sort of reanalysis that you're seeing down here at the bottom, if you can see my pointer. We do think a lot about gene discovery, and I kind of started this by telling you that we have a lot more to understand about genes. So while this isn't our only kind of bread and butter for our program, it is something that I sometimes like to kind of step back and look at what have we added. So far, we've identified, by my count, 95 novel disease genes and candidate disease genes, 12 in particular for which we've, in the blue, identified phenotypic expansion, where the gene was previously known to be associated with a human disease trait, but now we understand more about the range of phenotypes associated with that gene, and then many examples of sort of known genes with either novel variants or novel variant types. And with that, I want to go ahead and jump into a kind of an example, or maybe a couple of examples of what I would consider sort of a little bit of the bread and butter of our approach right now. So I'll talk first about DHX9 and neurodevelopmental disorders. This is work that's been led by Dr. Daniel Callum here at Baylor College of Medicine. He's by training a pediatric neurologist, but also very much a geneticist in the sense that he works closely with our group, so he can't get away from us. He reported 15 families with de novo DHX9 variants, and these were all ultra rare variants. They were putative loss of function or missense variants, and you can see the families here. What was interesting as he started to kind of put this cohort together is that he saw an additional 3 families that had a bit of a different phenotype. They had Charcot-Marie-Tooth neuropathy rather than neurodevelopmental delay. They also had de novo variants in DHX9. The variants were also ultra rare. They tended to be missense. And so he started to kind of put this together, and he looked at the variants across sort of the linear protein structure of DHX9. You can see here that many of these are missense variants. They really were located along multiple different regions of the protein. There are some putative loss of function variants, like this stop gain right here. And there were a subset of variants, actually 2 very specifically, that were identified in the nuclear localization signal of this protein. And he collaborated with Shen-Gu. Dr. Shen-Gu is one of our former trainees and now close collaborators, to try to understand what all of this meant and whether he could begin to piece together a genotype-phenotype relationship for these individuals, so that we could start to answer questions for newly diagnosed families, like what should we expect? What should they expect for their family member who has a variant in this gene? Should they expect Charcot-Marie-Tooth neuropathy? Should they expect neurodevelopmental delay? Might it be both? In doing this, he identified, he and Dr. Gu identified sort of a few different really interesting examples. And this is really done by combining both the phenotype data from the patients and understanding that in as much depth as possible, then also looking at the functional impact of these variants in cells. And so I'm going to show you sort of a few examples of what they learned in doing this work. For several of the variants that were associated with severe neurodevelopmental delay, actually the 2 that I showed you that were impacting the nuclear localization domain, the nuclear localization sequence, they saw that these variants resulted in the DHX9 protein being aberrantly located to the cytoplasm. And so you can see here sort of the EGFP backbone, sort of a control, if you will. This is one of those variants, and the GFP is lighting up the location of the protein, and the protein is not in the nucleus, not in the nucleoli, but really everywhere else. It's in the cytoplasm. There were another subset of individuals with severe neurodevelopmental delay who instead showed something very different with their variants. They had increased R loop formation, and I should have mentioned that DHX9 is an RNA helicase. These individuals also had increased DNA double-strand breaks, and so this was visualized comparing the wild type, and I'm showing you here just one of these variants, by the gamma H2AX signal. And so gamma H2AX localizes to the sites of DNA double-strand breaks, and you can kind of see those lighting up right here in the nucleus. And so this particular variant that led to severe neurodevelopmental delay, it included these kind of functional observations here related to double-strand break formation. And then contrast that with the individuals and their variants who had Charcot-Marie-Tooth neuropathy. Again, showing you wild type up top. This is a missense variant associated with Charcot-Marie-Tooth neuropathy. The protein continues to be shown kind of highlighted in green here. And now you're getting nucleolar localization of this protein. So it's not all over the cytoplasm. It's not specifically in the nucleus, but it's in the nucleoli. So aberrant expression. So 3 very different cellular phenotypes associated with 3 different variants, and a variety of kind of phenotypic expression of this disease. And so this was a really important demonstration of how the functional work here can help you begin to understand variant impact and perhaps start to predict variant consequences for the individual who has that variant. So I want to kind of step back a moment and just sort of simplify this story to the basics of what I just showed you. We talked about cohort generation. We talked about developing a human allelic series. We talked about deep phenotyping of individuals, identifying a phenotypic range, if there is one. There may not be, but many times there is. And then functional analysis to try to understand how different variants might lead to some of the distinct phenotypes that are being observed. It turns out that there are many examples within our work that essentially involve these iterative steps to discovery. And I'll highlight just one more that's unpublished, just to kind of show you why we think that this approach is so important. This comprehensive approach is so important. So the next story I'll tell you is FLVCR1. This, again, was led by Daniel. And in the interest of time, I'll move forward a little bit faster. But really identifying individuals who had one of two different phenotypes, some with an adult neurodegenerative phenotype and others with a severe earlier onset developmental disorder. And those are kind of represented by the different colors here. You can see sort of an allelic series. And again, this was a really great example where there were essentially missense variants across a majority of the linear length of the protein. And FLVCR1, by the way, is a choline and ethanolamine transporter, which we'll keep in mind here. The phenotypes, which I sort of already mentioned, adult neurodevelopmental versus pediatric, and you can see just a small smattering of that here. I'm sorry, adult neurodegenerative versus pediatric neurodevelopmental, and also some skeletal and physical findings. And then critical to what FLVCR1 does as an ethanolamine and choline transporter, can actually take advantage of that and look at the activity of that transporter and how different variants impact that transporter's activity. And so you can begin to put sort of activity levels, assign activity levels to each variant, and really begin to try to pick apart what is driving the different phenotypes that we're seeing in the humans who have these variants. So I really wanted to kind of show this mostly from a big picture standpoint to kind of highlight a lot of the bread and butter work that we do that essentially really involves these 4 steps and not necessarily always in exactly this order. I'm going to move on and transition just a little bit to something that is a bit near and dear to my heart, which is the identification of dual molecular diagnoses or multiple molecular diagnoses. So we know in the rare disease world that there are some individuals for whom lightning metaphorically has struck twice, and they actually have 2 different, sometimes more, genetic causes of their set of phenotypes. And so what I'm showing you here is actually kind of a little bit of a complex scenario. This is an example of a patient that came through our diagnostic lab. And this is an individual who has developmental and epileptic encephalopathy, but has damaging variants actually in 2 genes related to this condition. And this person actually had a much more severe course than you might expect if they had a damaging variant in either one gene or the other. So the individuals who have dual molecular diagnoses, they really have variants in 2 distinct genes, and they can have a blended phenotype that incorporates features from both of the conditions that they have. We know that accurate diagnosis for families is critical. It helps tell them and tell their physicians what surveillance management is needed. It informs recurrence risk for the family so that they understand, is there a risk to the next generation? Is there a risk if we have additional children? That can be very helpful information. And the fact that there are individuals out there who have more than one molecular diagnosis tells us that often the diagnostic odyssey for a family may not end with the identification of the initial diagnosis. So something to keep in mind. Many years ago now, we were very interested in understanding how common it was that families would have more than one molecular diagnosis. And we looked at this in collaboration with the diagnostic lab, Baylor Genetics here. And this is work that I did with Tamar Harel and Magda Valkiewicz. And at the time, the molecular diagnostic rate, this was for an exome sequencing cohort. These were clinical referrals. They were non-cancer referrals. The molecular diagnostic rate was around 28%. And if you took those 28% of individuals, it's around 2,000. Almost 5% of them had 2 or more molecular diagnoses that were related to the phenotype for which their physician ordered the testing. You can see most people here, 97 of them had 2 molecular diagnoses, 3 of them had 3, and 1 had 4. We've been really interested in a variety of components of this ever since this observation and this study, and recognizing that there's more than just a handful of people out there who have multiple molecular diagnoses. One of the things that we've looked at is what are those combinations of molecular diagnoses when you do see them? And in the diagnostic lab cohort that I just mentioned, it's primarily a North American clinical referral cohort. Individuals most frequently have 2 autosomal dominant diagnoses, but you see other kind of combinations, dominant and recessive, dominant and X-linked, 2 recessive, et cetera. And so you can really look at this and start to understand, for the individuals who do have 2 molecular diagnoses, with this information about Mendelian inheritance patterns, what the recurrence risk for each of those diagnoses would be for the family. When we compare this, though, to a Turkish cohort, we have a rare disease cohort that's part of the former Baylor Hawkins Center for Mendelian Genomics and has grown in the Baylor-GREGoR program. We see that the rate of dual molecular diagnoses, first of all, they are identified, but there's a really fascinating shift towards combinations of 2 recessive diagnoses. So these are individuals who tend to have biallelic variants, either compound heterozygous or homozygous, that are leading to both of their diagnoses, and sometimes even combinations of 3 recessive diagnoses. We've been very interested in thinking about what's driving this. We know that these families most frequently will report some degree of parental relatedness. So we know that there is a high degree of identity by descent compared to a North American cohort. And if you look at the frequency overall of multiple molecular diagnoses in this Turkish cohort, if you limit that to just known genes, kind of thinking about how clinical laboratory reporting often follows, we're talking about 12.2% compared to the almost 5% I mentioned earlier. So it's a much higher rate of dual molecular diagnoses. And that fraction goes up much higher if you include all diagnoses, including candidate diagnoses. And of course, then the rate of 2 recessive diagnoses is much, much higher in this group as well compared to a North American cohort. And so we know that one of the things that contributes to the rate of multiple molecular diagnoses is the presence of identity by descent in some rare disease families. This has really driven us to focus in on unique populations, and one of them being the Turkish population. So we've been very fortunate to have over the years several Turkish physicians time with us, mostly geneticists who spent time with us embedded in with our team, referring patients to us, and then making connections with other referring physicians who've remained back in Turkey. And so we've identified or developed a cohort now of over 1,400 unrelated Turkish families. Through that, there have been, and I'm sure this number has probably gone up since the last time I updated the slide, at least 138 novel disease genes, at least 15 phenotypic expansions, and at least 78 families for whom there were multiple molecular diagnoses. With this size cohort, one of the really critical things that we're able to do is to start to ask questions about how admixture and consanguinity, parental consanguinity, can shape the molecular pathogenesis of human disease. And so this is work I mentioned Dr. Koban Akdemir at the beginning of my talk. She's over at the School of Public Health at the University of Texas next door. She's been leading this and actually is probably actively looking at the galley proof for her paper, which I've listed here, that really describes this and trying to understand admixture and consanguinity and how that contributes to molecular diagnostic rates. One of the really fascinating things that she's found is that if you look at Turkish individuals on the whole, they, first of all, it is known to be an admixed population, but there really are two very different clusters of individuals within that. And so she's been exploring what that means for the population, what that means for diagnostic rates, what that means for multiple molecular diagnostic rates, and the rate of absence of heterozygosity. She's also developed a population-specific database for variant minor allele frequencies from this Turkish cohort specifically, so that individuals who are studying families from Turkey, which are largely underrepresented in population databases, if you want to know if a variant is present in our Turkish cohort, you can look that up here. And I'm happy to share that link after the talk as well. So all of this, I think, success and early discovery with the families that we've worked with from Turkey has led us to really dig into that region as part of our recruitment. We've been very fortunate to develop collaborations with physicians throughout the Middle East and North Africa, and have really developed a cohort there. And there are, I think at this point, a little over 306 unrelated families from this region, about 86% of them reporting parental consanguinity. And a lot of this work has been led by Dr. Dana Marafi, who's a pediatric epileptologist. She trained here at Baylor and is now back in her home country in Kuwait, practicing medicine, continues to be a very active collaborator. And she's also worked very closely with Vince Duan, who's a former student, now postdoc, here with our group. And this was a nice slide that Vince put together, kind of looking at how often we were able to recruit from afar trios or quads, where we had a proband who was the affected individual and both parents, typically unaffected parents, or the proband, a sibling and both unaffected parents for study. And you can see a majority of these families were actually able to get a pretty good trio or quad that we could then study to identify both de novo variants, but also compound variants. We certainly don't limit our recruitment to trios and quads. So for families where maybe only the proband sample is available biologically, or only a duo, the proband and one parent, of course, we accept those as well, and we accept larger families. This was just sort of mapping out the distribution of the family size and the individuals we've been able to sequence and study. The overall molecular diagnostic rate to date for this cohort has been around 45%. And there's analysis that remains ongoing. And I won't kind of belabor these points, except to kind of reinforce the point that I think there's a large fraction of individuals who have recessive conditions resulting from homozygous variants, but you do see sort of a collection of other inheritance types and other, and as well as recessive related to compound heterozygous variants. And so it's very important to really consider sort of this full spectrum of possibilities when analyzing data from these families rather than looking only at the homozygous variants. A really great example of a study that's come out of work in this region, also led by Dana and Vince, is the story of LGI3. So together, Dana and Vince assembled a cohort of families with peripheral nerve hyperexcitability syndrome. They were able to generate this cohort in part through recruitment to our center, but also through gene matcher and matchmaking programs to identify additional families. And what's really amazing is that they were able to identify families from really all over the Middle East region, as well as some other locations. And what was really exciting about this finding is they were also able to partner with researchers to make an LGI3 knockout mouse to really understand what was driving this peripheral nerve hyperexcitability syndrome. And what they found is that the mice that lacked the LGI3 protein, first of all, their electrophysiological studies were very similar to what was seen in humans, but they could also dissect the peripheral nerves of these mice and see that the KCNA channels that are so important at the juxtaperonodal region were mislocalized. This was critical because we know that LGI3 co-localizes with these channels. And when you don't have LGI3, the channels don't localize where they're supposed to. And so the knockout mouse had a very different and far less functional juxtaperonodal region. So I'm going to circle back for just a moment to the dual molecular diagnoses, but this time with a slightly different focus in hand. And I see that I have 20 minutes, so I'm watching the time. Talked earlier, when I showed you this example, I talked about an individual who had two different genes, variants in two different genes contributing to developmental and epileptic encephalopathy. So two genes contributing very similar disease traits, very similar sets of phenotypes, if you will, the developmental delay and the epilepsy being key for these individuals. We know that there are individuals, though, that can have dual molecular diagnoses where the two conditions are actually very different. And so on the left, I'm showing you an individual with oculocutaneous albinism and Loeys-Dietz syndrome. And so you can kind of quickly get a sense the phenotypes for oculocutaneous albinism are very, very different from the phenotypes or the features that are associated with Loeys-Dietz syndrome. And if this person were to walk into a clinic, you could envision someone saying, I've not seen this combination of phenotypes before, you must have a new condition, when in fact, this person really has two very well-known conditions. And so I want to kind of set up this idea that there are dual molecular diagnoses where you can have distinct phenotypes, distinct sets of phenotypes, and some where you can have very overlapping sets of phenotypes resulting from those two diagnoses. And we can start to think about how to computationally measure how overlapping or how distinct these two sets of phenotypes are for individuals who have dual diagnoses. And I will build on this, but we very early on, as we started to think about this, felt that it was very, very important to be able to do this in a computational and objective way. And so our first stab at this, now many years ago, was to take the individuals that we had identified through the diagnostic laboratory collaboration and assign them as either distinct, meaning that they seem to have two very different conditions contributing to their phenotypes, or overlapping. And these assignments were made by two physician scientists. And then we asked the question, can we set those assignments aside and computationally model these two disease pairs for each of these individuals and ask the question, if we generate a phenotypic similarity score, where we take an individual, their two diagnoses, and we ask how similar or dissimilar are these sets of phenotypes? What can that tell us? Can we use that to begin to predict who might have a distinct pair of disease traits versus who might have an overlapping pair of disease traits? And can we do this using the human phenotype ontology computationally? And in fact, it turns out that you can. So if you calculate phenotypic similarity scores for these disease pairs that we observed in real individuals who had clinical testing through the diagnostic lab, we see that the individuals that were assigned as having distinct pairs had a lower phenotypic similarity score than those who were assigned to have overlapping disease pairs. And so this was kind of really the start of us asking questions related to what more can we do with this approach to better understand phenotypes. Now, I'm going to show you a small part of that here today. So the next question we asked, one of the next questions we asked is, can we take advantage of the ontological structure of the human phenotype ontology to perform pairwise comparisons of individual patients and their phenotypes? So now moving away from dual molecular diagnoses, just asking, can we compare pairs of patients? And for those of you who are not familiar, the human phenotype ontology, it is an ontological structure. It kind of starts at the top, if you will, with a phenotypic abnormality, and then you might have abnormality of the nervous system shown here in teal, abnormality of the musculoskeletal system shown here in green. So your first branches are really organ systems. And as you move down the branch, your terms become more specific. They have more information content. So abnormality of the central nervous system is not as meaningful as abnormality of the cerebellum. But it's very clear from this structure that those are two related terms. And so the question became, can we use this to compare individuals? And not only that, but can we use this to compare individuals who all have the same condition? So what we're talking about is taking two individuals, let's say patient 1 and patient 2, who each have condition A, and they have a list of phenotypes. They have a list of clinical features. And each of those features they either do or don't have. And can we compare those two lists and generate a phenotypic similarity score? And if we do that, what do we see? So this is work that was very much led by Claudia. And the focus was on Robinow syndrome. So the patient condition was Robinow syndrome. And this is a skeletal dysplasia with characteristic clinical findings. And so the clinical diagnosis for these individuals was not in question. And the subjects that we studied at the time, there were 68 of them, had all been very well characterized here. And actually, many of Claudia's colleagues will probably recognize many of these genes in this pie chart are genes that she, in fact, led the discovery and description of. So you get the sense that there was genetic heterogeneity in this cohort that we studied. But again, they all had Robinow syndrome. And we took each of those 68 subjects and we did this comparison and generated pairwise phenotypic similarity scores for every possible pair of those 68. And we got a heat map that looked kind of like this. So as you might expect, down the diagonal, you have red. This is self to self, so a phenotypic similarity score of 1, the highest possible score. The bluer color representing, or the colder colors kind of representing lower phenotypic similarity. It's a little, I should say, we took those similarity scores and then performed cluster analysis as well. So then we asked the question, what clusters together? And what was absolutely fascinating to me is that the individuals who had autosomal dominant Robinow syndrome due to DVL1 and 3, disheveled 1 and 3 variants, largely clustered together. Individuals with truncating variants in frizzled 2 clustered together and separate from the disheveled group. And then there were a couple of missense clusters for frizzled 2 that clustered within the DVL1 and 2 clusters. And what this taught us is that we could actually take this computational approach of examining phenotypic similarity, really use it to start to understand more about the relationships between distinct genes and disease traits like Robinow, that's genetically heterogeneous, but potentially also different variant types within some genes and how that might influence human phenotypes. So now things start to get really exciting, and we go back to one of my interests. We asked the next question was really, can we use human phenotype ontology and this phenotypic similarity approach in some ways to dissect an individual who has multi-locus pathogenic variation? So this is a young child from Egypt who had very severe neurodevelopmental delay. And the question arose, well, I should say, first of all, his exome was analyzed by Isabella Herman and Gad Jolly, AJ, both of whom have since moved on. And they came together and said, we really think that there are 4 candidate molecular diagnoses here. 3 of these are in known disease genes, so ZC4H2, Weicker-Wolff syndrome, MuSK, which is congenital myasthenic syndrome, CAPN3, limb-girdle muscular dystrophy syndrome. But also there's this compelling variant in a gene called NAV2. It's associated with cerebellar dysgenesis in the mouse. No human phenotype had been reported at the time. The question kind of became, first of all, it's impossible to publish this, but how do we even convince ourselves that these variants are all contributing, or are they? Maybe they're not all contributing. So what do we do to sort this out for ourselves? And then, of course, how do we get this out there into the literature? So the first thing that we tasked AJ with doing was to really make a list of all of the different phenotypes that this particular patient had. And so BH14188 is our patient. These were all the clinically observed phenotypes, marked in red because they were all present in him. Then we asked the question for the 3 genes that had known human disease phenotypes, how well did these kind of answer or not answer the different phenotypes that we knew were present in the patient? You can see some that to some degree, some of the phenotypes maybe we could explain, others a little bit less clear. And if you just looked overall at the combination of these 3 known human disease genes and the phenotypes they might explain, you got this sort of synthetic clinical phenotype that you might predict from these genes. It looks something like this. But you can see here a lot of gray boxes. These were phenotypes that remained unexplained at the time. And so we went back to the NAV2 question, the NAV2 gene, that 4th finding, and asked, what might that bias? And AJ was able to take advantage of HPO2Go and the existing mouse knockout NAV2 phenotype information and predict human phenotype ontology terms and circle back around to this and show here in the gold potential phenotypes in our patient, observed in our patient, that might be explained by NAV2. And you can see that there were several phenotypes not explained by the combination of the other 3 genes. And so the end result was that, or the conclusion that we had from this was that all 4 genes and their variants were contributing to this particular patient's phenotype. So we were able to use human phenotype ontology and really, I'll argue, very detailed, deep phenotyping that was obtained from the family and the physician to be able to put this together. Okay, I think I'm on my last story, and I'll go a little bit faster so that we have some time. So I want to talk a little bit about some work that was led by AJ and Gad Jolly on Merck's Meyer-Rokitansky-Kuster-Hauser syndrome. This is a congenital reproductive disorder in women. They tend to have an absent or underdeveloped uterus and vagina. They're often not ascertained until in their teens because they may simply present for the first time with amenorrhea. They can have a type 1 or a type 2. Type 1 is isolated, type 2 is syndromic. And AJ worked with several OB/GYN specialists to assemble a cohort of primarily US-based and European individuals and performed a rare variant enrichment analysis in these individuals from their exome data and identified variants or identified GREB1L as enriched in these individuals. And we kind of turned to AJ and said, well, let's look at what's known about GREB1L. It's been associated with congenital kidney malformations before, bilateral kidney agenesis, renal agenesis. We weren't initially completely convinced. And so AJ was tasked with the very, I guess, undesirable task of really mapping out all of the variants that had ever been described before in GREB1L and mapping them to the different phenotypes that had been described, either in the cohorts that he had developed or those that had been described before in the literature. And so you can kind of see he's in his legend, there's female reproductive tract malformations, urinary tract malformations, skeletal and digital malformations, and ear and cranial nerve 8 malformations. And I show this slide because I'm so impressed at how he organized all of this information. It's tremendously useful if you're a physician who's looking at a patient wanting to kind of map out your variant and where they fall and whether it's been reported before and what that might mean. But it's very difficult from a presentation perspective to take anything from this. And so AJ also mapped these out in a very different way along the sort of exon structure of the gene, but also by whether the variant was loss of function or missense, or in this one case, an on-frame shift variant here in green, and then mapped out the different phenotypes that were observed. And what you can quickly gain from this view of the data is that there's some exons where very clearly the phenotypes were only female reproductive tract malformations. There were other exons where you really would see sort of a much larger combination of many of these different features in the individuals who were reported. So starting to give us a little bit of a glimpse into how to understand genotype and phenotype relationships with this gene. And kind of the output of all of that is that it became very clear that while there were absolutely female individuals who had reproductive tract malformations and only reproductive tract malformations, there were a lot of individuals with GREB1L variants who had varying combinations of these different phenotypic features. And with that, I know we're towards the end of the hour. Go ahead and wrap up with conclusions. I hope that I've convinced this group today that we have, an increasing or continued need for discovery, disease gene discovery, variants, understanding how variants impact phenotypes, understanding molecular mechanisms underlying those variants and those genes. I hope I've convinced this group that a comprehensive approach to molecular diagnosis and discovery is critical. So those steps that I talked about earlier, not necessarily in a stepwise order though, that cohort building, generating a human allelic series, understanding deeply as much as possible the human disease phenotypes, and then functionally investigating the variants within that allelic series and trying to link those functional impacts back to human phenotype data. And then finally, the human phenotype ontology as an objective and computational way to assess human phenotypes. And I showed you examples of how to characterize dual molecular diagnoses, how to investigate phenotypic expansion, how to identify or test multiple molecular diagnoses and those types of hypotheses. And with that, I'll go ahead and finish. I'd really like to acknowledge and thank our wonderful team, and of course, Claudia's team over here, and most recently, I would say we've had some great interactions with Jeff and Cliff, and kind of my funding as well. So thank you all. Happy to take questions, or I guess we might move on to the Q&A.
Jen, thank you so much for this wonderful genetic class. I wanted to start with the questions to you, and then we can get Jill back if people want to. We can start the discussion together. Any questions from the audience to Jen? Amy?
People are probably still typing.
So, Jen, I have a question because you showed a lot of very interesting examples of genes that have they cause multiple phenotypes, right? Depending on the mutation. So obviously, allelic series is a very interesting way, and I guess relevant way to discover that. Do you have any idea how many genes and what type of genes do you predict to have this type of behavior?
Ooh, that's a really good question. That's a really good question. So first of all, without a doubt, I think that they're unrecognized. And I think that they're particularly unrecognized because if a gene is first associated with one condition or one set of phenotypes, often when variants in that gene are encountered later on in a different scenario, perhaps analyzing or looking at data from a family that has a very different phenotype, people will almost quickly kind of discount it and say, well, this isn't a phenotypic match, so it must not be these variants. And that can be kind of limiting thinking. But because of that, I don't think we have a good sense of this. And related to your question, there are a lot of genes that we see now that are associated with conditions that can demonstrate both dominant and recessive inheritance. Some of them, it's the same condition, either dominant or recessive. For others, it's very different conditions that are dominant or recessive. And I mean, lamin A is kind of one that, right, can give you headaches and the different phenotypes and modes of inheritance that are possible there. And I think that's a great example of the possibilities out there. But in terms of a true assessment, I don't know. Years ago, I looked at the OMIM data and looked at how many genes were associated with more than one sort of disease entry, more than one, I guess, hashtag entry, if you will. And I believe at the time I looked, it was around between a third and a quarter. And I would have to envision that is increased. Okay. Yeah. Yeah. So I don't have a good answer to your question, but I think we're missing a lot.
Yeah.
And I'm interested in these patients with multiple mutations. Do you think those are additive interactions of those mutations, or do you think that they're sort of non-additive specific interactions that only manifest in the context of those two mutations in terms of the phenotypes that you're seeing?
That's a really good question. For the genes where I guess my short answer will be I don't think it's been across sufficient number of cases to fully know. I think that there are some where it's definitely additive, but I'm very much compelled by the idea that there are some scenarios where it's much more than additive. And you can think about, I have to say this kind of carefully, you can think about digenic conditions, which don't segregate as two independent conditions, but where often your gene pair, they have an epistatic relationship, right? They're often either in the same pathway or both of the protein products are part of a shared complex or things like that. They are interactors in some way. And I think the digenics are almost an extreme of that, where you require both to have phenotypes at all. But that was kind of a long answer to your question, but I think there's probably some variations on that, and it's really tough to understand and to probe the scenarios where it's more than additive, simply because each of the humans we study tends to be an N of one. And so it's rare to see that same combination over and over again enough times to really be able to probe what's going on phenotypically.
Thanks.
Question. Hi, Jen. This is Jill. Thanks for that presentation. Sure. Yeah.
So I'm wondering if this type of analysis has ever been done where, you know, I'm sorry, and this is just when I think I've mastered some science, I hear a presentation like this and I'm like, whoa, there's so much more to learn. So the multiple kind of variants is fascinating to me, and seeing sort of what kind of overlap this was. In the case of our disorder, where most patients have a sibling, an affected sibling, due to it being autosomal recessive. So they have the same variants, but yet we're seeing quite a bit of heterogeneity, even within a family. And the families are asking about this. And like, okay, well, you're telling me they have the same thing. Why are my kids so different? I mean, compared to the general population, people even say that to my kids, like, are you sure they have the same thing? And I'll say like, well, they check all the same neurodevelopmental boxes. You know, within that, there's a range, you know. So I guess, has anybody used this type of analysis to look at maybe protective genes that might make a light version of a disorder within a family?
That's a really interesting question. We have a close colleague who's actually looking at that in a very limited number of conditions. One of the challenges, although it's different for FAM because of the biallelicness of the variants, and so the fact that you have SIV ships where there's more than one. But one of the challenges is often because sometimes there's a relationship between what is the exact variant and the phenotype. The best way to test that is to amass many people who have the exact same variant and then start from there and go, who's similar and who's not similar? Are some people milder? And then do they seem to have something else that now we can find by comparing their data? So it's a really, really good question. We have started to ask, more than started, the opposite question, which is, are there things that make some people more severe? This is work that Jim Lupsky has led. His interest in Charcot-Marie-Tooth neuropathy, I think, is a really great example. And it was born out of this frustration that he and many others have had in the clinic, seeing individuals with Charcot-Marie-Tooth. So there's actually a recurrent copy number variant that causes a high fraction of cases, so that you have families where multiple families and multiple family members all have the same variant that's causing their Charcot-Marie-Tooth neuropathy. And yet when you meet with these families, it's impossible to predict when someone's going to develop their neuropathy or how severe it's going to be, because within the same family, so sharing kind of the same genomic background, the same variant, you get this wide spectrum of severity. It's all neuropathy, but this wide spectrum of severity. And so what he looked at was whether or not there were variants in genes known to be important for nerve function, known to be associated with neuropathy, but variants that by themselves wouldn't create any problems. You or I could have them, we'd be fine. But that when you pair those variants with this neuropathy causing variant, if you will, that suddenly the phenotype is more severe. And in fact, you can demonstrate that. And we refer to that as a mutational burden. So the opposite half of your hypothesis has definitely been demonstrated, and at least for neuropathy in a few conditions. And we think that this is happening much, much more than we yet fully understand. The challenge to this is amassing a number of individuals with the same condition so that you can really compare across variants and phenotypes and within families and across families. But your question's a really, really good one, and it's something that we think we need to better understand.
Great, thanks.
Okay. More questions for them?
I actually have one myself. I didn't see any other questions in the chat. So with respect to kind of ultra rare diseases, right? So we have an example where, there are 33 patients in the literature and they've been analyzed to the extent that they can be analyzed. And then, we have a complete mutational scan and information about the functional impact of those variants plus all the other ones. But there's this disconnect between, kind of what is the full phenotypic spectrum? And I'm wondering what your thoughts are we now entering kind of the golden age of basic old-fashioned biology where, so for example, kind of what might be needed is, a really deeply phenotyped mouse model, for example. And that's to some extent, kind of really old-fashioned biology, but that's really what's holding up progress for this particular disease. Do you have examples from your own work where that type of really deep phenotyping, if it's been done in some kind of mammalian model system, can really kind of, synergistically allow you to do things that you couldn't have done with a limited set of patients with ultra rare diseases?
That's also a very good question. There is always and will always be the caveat that sometimes the mouse model doesn't perfectly recapitulate the human phenotype. And so that's a bit of a challenge, because when you see a phenotype for the first time in a mouse model, until you've seen it in at least one human individual, hard to know if that's just going to be mouse specific or if it's going to be something that we see in people as well. Because of that, one of the approaches that we have been very interested in taking and try to take is what we kind of refer to as a gene first approach. So to kind of your point, the power often in these ultra rare conditions and studying them is to really develop a cohort of as many people as you can that all have rare variants in that same gene. And then you can start to ask everybody, well, what is your phenotype and what is yours? Really deeply compare. So sometimes with some of these smaller kind of case reports that come out, it can be really difficult. We're very interested in when we identify the gene, trying to work through gene matching programs really worldwide to identify as many people as possible that and from many different populations, many different places in the world, which can sometimes be important when you think about phenotype expression and kind of genetic background. So that we can really understand as much as possible what that sort of range of phenotypes in humans can be. So kind of use the mice to compare, but you're always left with that little caveat of the mice don't always equate to what you see in humans. And it's particularly complex when you start looking at variants that we have that are not complete loss of function variants, where they probably don't equate to the variant that we might be making for the knockout mouse. There's a lot of work that's starting to be done tailoring the mice so that maybe if someone has a missense variant, can we make that exact same variant in the mouse? And if that mouse's sequence is a little bit different, can we essentially sort of humanize it so that we're making really the exact same protein that our patient has, and then seeing what happens in the mouse? We do this also in flies. And I think that may get us a little bit closer, but there's several layers of complexity here. I think it's always going to be a partnership between putting together as much data as you can from the people who have the condition, but then also kind of thinking really smartly about what are our takeaways from animal models.
I think there are the opposite as well. Like sometimes the mouse models can help predict that the disease exists, right? And I guess
that
the
for sure.
medication is one example of those. Buddha made the mouse, the duplication mouse before of the disease has been really seen in human. I guess there are maybe Justin actually has a paper on this. I think thousands of genes in this situation. Okay, I have a question for both of you. Like, in your opinion, separated, what do you think is the most important thing that in rare diseases we need? Like, in terms of either research or in terms of community building, in your opinion, John, what's your
Funding.
Yes.
We need to identify patients sooner, get them connected to communities, collect data in a really smart way. And keep putting out really brilliant researchers like yourself and get them interested in studying rare diseases.
I agree with all of that. Oh gosh, what can I add? I think the one other piece that's really critical is just the kind of data sharing component. So collecting all of that data, the clinical data, the phenotype data, the genomic data, and then making it available for folks to analyze, because more heads and more folks trying to tackle the same problem is better than one group. And so increasingly, really, I think a lot of the databases like Anvil and things are great, and the next step may be to find ways to sort of fenestrate those so that someone could really go in and say, I want to look kind of across the world's cohorts. This is pipe dream, right? Identify everyone who has this variant in this gene, and then pull out their phenotype information and see if it all makes sense. We're probably a little ways away from that, but I think that a lot of the data sharing and things is going to be critical to making that happen and moving forward the science more quickly.
And then I will go back to the idea of open science and collaboration. Patients first, then publish.
from the standpoint of a very much of an outside observer, it appears to me that the salience of rare diseases has, in the broader public, has come a long ways in the past, say, 10 years. I wonder if that's just me or whether you would confirm that from your much more central positions.
It's certainly more relevant to me, and it's hard to say how biased I am because we suddenly got a diagnosis, we have a target. When I said I said we wanted a community, and I assumed we needed a diagnosis to get that community. And what I wish I had realized sooner is that we actually didn't, that we belonged to this broader community of rare diseases. And I don't know if I missed it in the early days, or to your point, it's become far more relevant in the last 10 years. Maybe both. But, so there's awareness campaigns that are, and there's consortiums of rare disease groups and think tanks that have really accelerated the missions of all rare diseases. And I think it's probably all the modalities that exist now to advance the science and the sequencing. Like, these are actionable diseases now. So I think that the relevance of rare disease is increased because of that. Hopefully there's going to be fewer and fewer families that get a diagnosis and a doctor says, and there's nothing you can do, because it's just not true anymore. So that's all.
I absolutely agree. Go ahead. Oh, okay. I think we spoke at the same time. I absolutely agree. I think that adding to that, I think that there is a better understanding in general about genetics and genomics than there was 10 years ago and what that can mean for health. There's imperfect, but more availability of broad testing like exome and genome sequencing. So people are a little bit more likely to get diagnoses than they were before if they have a rare disease, and to achieve those diagnoses sooner, although certainly not yet fast enough. And so I think some of these things are also conspiring to kind of just increase the general awareness of this. I think it helps when people can say, yeah, I have a neighbor who has something kind of like that and it's genetic. And I think the number of people that can kind of say that now is a lot higher than it was a decade ago. So the awareness has increased, and I think that's a very good and important thing.
The other thing I want to comment on, and you maybe picked this up in my talk, is the roadmap is not linear, and that we can move to translational science right away, right? And I didn't understand that. I didn't even understand what translational science was, right? I just thought like, well, we don't know the mechanism of this disease. There's really nothing we can do yet. And that's not true either. And our fly screen demonstrates that. Like, it's possible that we are going to find a therapeutic, a drug that improves our kids and have no idea why. And that's okay. We can reverse engineer it. We can figure that out later. So all of these things can happen at the same time. That's pretty exciting.
For both of you, and I think I've got some of the answers to this already, but what one thing, one idea, one fact would you most want members of the general public to know in the arena of rare diseases?
I would say that we're worth investing in. The common diseases are important to invest in too. But in general, these are really, really well-funded diseases, really well-funded. And in some case, they might not have a single cause, a single target. That's why they're juggernauts. That's why we haven't solved them yet, right? But in cases of rare disease, you saw the number was that 72% have a genetic cause. Okay, that means we can figure it out and potentially we can fix it. So the return on your investment is potentially very high without a lot of investment. So I would say, like, we are a great investment, rare disease. And it might inform the bigger diseases. It probably will. There's a lot of shared learnings, even across rare diseases. And there's work being done to find the overlapping phenotypes and the underlying mechanisms that are the same. And, you know, don't underestimate these rare disease warrior parents. Like, we're going to figure this stuff out because we have to, because other people aren't.
I fully agree. I mean, I would have two thoughts, and really it's very much encapsulated by what Jill already said. Rare disease collectively is not really that rare, number one. And then number two, I think that not only is rare disease research important for all of the people that do have rare disease, but we're absolutely starting to learn a lot about some of the biology underlying more common disease. And that's in my mind going to be very much a spectrum. So these lessons are applicable to everybody. This will benefit everyone.
I think there is, so Lisa wants
This
to.
is steps and what I can do. Lisa, do you want to go ahead and unmute and?
Hello, thank you. Really, your talks were wonderful and very inspiring. And I'm sorry I can't be there today, but I wanted to just put a little thought in everyone's heads and about that relate to what both of you have been talking about. One of the things is that, for example, my group works on neurodevelopmental disorders using mouse models, and mouse genetics is quite far along because you can so easily pair up animals and measure phenotypes and so forth. It's a lot faster and easier, and think you can do things with it you can't necessarily do with human patients. But what you've all been talking about just brings to my mind that there are ways to think about rare diseases where they are not rare. That is, your rare disease might be part of a spectrum of diseases that overlap. And Jennifer was showing some cases where if you look at the diagnoses, they overlap. That tells you, as a mouse geneticist, I would say, that those genes might be interacting in similar pathways. And that what's happened is that they are interacting in some way and supporting each other in a way that make the disease more likely to happen. So what I want to say is that FAM 177A1, I think, is possibly part of one of those groups. And if you think about, especially from the point of view of neurodevelopmental disorders, if you look at autism, it's not one gene. It's a whole bunch of genes. And every case of autism is a unique rare disease in some ways. They don't all share all the same phenotypes. They're just classified that way because altogether they're common. Individually, they're not. So if you start to think of your diseases in terms of being part of a spectrum that overlaps with other diseases, some of the cures that may be available for those diseases, or that may come available from those diseases, could be relevant to you. And if you pile all the different rare diseases together in groups that do share diagnoses, together they may help drive a cure. Just a thought, because it's certainly the way things have gone in the neurodevelopmental disease research in the past, even for these common disorders like autism. It's really just a bucket of diagnoses that converge on some social problems. These are all kind of rare diseases that together make a common disease. And you can think of your rare disorders in that way in some respects too, I think. And that might help you find a cure.
Excellent point. Thank you, everyone. Next month, we have John Ray with us. So thank you for coming.
Thank you.